Good morning. Thank you very much for the possibility to talk here. Um, well, I'm doing this intermix, so the slides are in Spanish and I talk in English, and hopefully that's fine. And what I want to present is a little bit more science-based, together with two collaborators, long-term collaborators, Vanessa Falcon from La Habana, who is also here, and Pedro Gonzalez from Origen in Cuba. And yeah, so the topic is, so what is the origin, evolution of the flora of Cuba and the Caribbean, and what do we really know? So, so as we all know, the Caribbean is one of the global biodiversity hotspots, and there are about 13,000 species of angiosperms, about half of them endemic in the region, but also for, well, if you talk about Cuba, depending on the group, it may be even more, but basically, yeah, this is what is also a very interesting pattern, and maybe we can consider sort of this region as a lab of evolutionary biology. And I'm just trying, well, I hope this is more science. I'm trying to not to go too deep into methods, but still, I think for having this clear, what are the major questions? So first of all, yeah, it's the big question that we all understand and have in our minds. What are the species and where are they? But then the question is, how did this evolve? So what are the patterns of diversification? What is sort of the time and space diversification? And also, what are actually the factors that have, since millions of years, influenced the evolution and species formation? So, in fact, as you all know, evolutionary biology nowadays, with the ease of sequencing DNA, has revolutionized what we're doing in taxonomy. So there is the field of integrative taxonomy that I will also talk about, which then is really the base to really have biological units classified and named as species. And of course, this is then also the units, the species that are needed as a scientific base for conservation status assessment. So this is a very similar or very simplified picture. Basically, we're using leaf material to generate, to reconstruct trees. This is a summarized tree of flowering plants as a whole, but the methods are very similar as we come down to the species level. And then if you're talking about geography, one issue is also very relevant. We need to see how do we analyze it. Essentially, these phylogenetic trees, so this is reconstructing the evolutionary history of our organisms through time. We can apply a molecular clock. We can calculate when which diversification event happens, but then we need to worry about geography and also, yeah, well, here is the Caribbean islands. The question also certainly is, what is the Caribbean biogeographically? But then, yeah, where did all the ancestors come from? And there have been various hypotheses through time. And I think this is one of the most important ones, sort of the Garlandia hypothesis. I think Santiago showed this land bridge in his presentation in the evening also, that was assumed to have having been there at the end of the Eocene. But the question is, would this be, when we know we can sort of calculate back diversification of organisms in time and space, would this also be confirmed with actual evolutionary biology data? So this topic has been around since 2004. There was the first review really in 2004. The broad analysis with molecules didn't really exist. And if we look at the literature, what is also quite interesting, which groups have been studied. So leukoproton, Brad, you're also here, so you will see your name up on the plate, is a genus which exclusively grows in Cuba. Then Spatelia also only in the Caribbean, and as you can imagine, we need to think about which taxa to sample in order to test these hypotheses. So this is also one of the earlier papers of a genus called Grünfelsia. Maybe you know it from gardens because it's a very attractive horticultural, or many of the species are very attractive horticulturally. And so here it was found that basically there are two clades, one Caribbean clade, one South American clade, but there are no Grünfelsias native neither in Central America nor Mexico. So of course, this is limited in testing really the overall biogeographical patterns. Now, things have been developed, and we have basically coming from collaboration between the Jardín Botánico Nacional in Cuba, the University of La Habana, and our institute, and others in the provinces of Cuba. We have, like 10 years ago, looked for what could be sort of adequate model groups to look at, because, well, you need to have the taxonomy, you need to have sampling, which has to be huge, sampling many, many species. So it's obvious that such deep analysis cannot be pursued with all the flora. It's impossible. So here is, for example, the Rubiaceae, Guettarda, Acalypha, this is Rubiaceae, Cocolova, this is a plant everybody knows, also the species here from the beaches on the region in Caparidastum as one of the examples. So this has been a program that we have been with PhD studies, also including collaboration with UNAM in Mexico, because we will see later that Mexico and Central America play a very important role in understanding all these patterns. So this is the model groups that we have been working with, and what I will show you and are still continuing. What I will show you is a little bit a wrap up of the results that were sort of obtained during the last decade. So this is one of the first comprehensively sampled trees. It's Euphorbiaceae, one subfamily, which is widely spread in the region, widespread in the region with many, many species. What we can also find in here, well, all the taxa, it's not really important that you can read all the genus and species names here, but what the pattern I think is interesting. So we, for example, have here this clade. There's not Leucophoton, Nauticloton, Garciladelia. This is the newly discovered genus from the Dominican Republic, and we see, okay, this is how you read the tree. Basically, here are we now. Then it has to be look back into evolutionary history, and then we have basically where the red arrow is located here, a so-called crown group where we can go back when like 10 million years ago, where we have reconstructed that this lineage has started to diversify. Now the problem, or the question is, what are the taxa? So we are widely sampling. Many of the genera are in the Caribbean, are in adjacent mainlands, as it would be called. And what we can see here, for example, all the red lineages, there are multiple origins of Caribbean endemic lineages. And what is also very important, we cannot really look at what has been the case historically a lot, what are sort of the endemic genera, because most importantly are the origins of species which have Caribbean endemism of very widespread genera. And then what we can also use these genes for is sort of reconstruct where the ancestors have been. So we can basically code distribution areas. Of course, this is not so easy to do properly at a species level, because we have to make sure that identifications are correct, that we have to make sure that we know it's going via the names, that all the plants are properly coded for. But what we can basically do here, also again, the methods and the details are not relevant for us now, but what the conclusion is, essentially, well, what are the ancestors? Where did the ancestors that arrived at the islands come from? And in this case, like 2/3 Mexico, Central America, but also there are some from the south, so coming in via the lateral hills, and this is what you can see here, very complex patterns that we can observe. Another group, which was the PhD project of Vanessa Falcon, is the genus Philanthus. So essentially, we can also find the problem was it's a pantropical genus, about 800 species, and that illustrates well. We have yeah, here's the Caribbean plate, here's another Caribbean plate. Mostly these are herbaceous taxa from Hispaniola, so Santo Domingo and Haiti. That's largely, not exclusively, but largely human radiation. So I think this underlines how important it is to sample and not too widely and not to have sort of pre-assumptions which at the end of the day are not really feasible. So of course we need to bring this in line here. This is the basic set of markers. So what we basically have been developing is a standard set of tested the nuclear markers, which is tied to everything. And then once there is a framework, we can go into more details using phylogenomic methods, because as we can also observe here, genetic distances between species that have been diversifying since very recent times, maybe very little. So it's a really massive challenge to do this well. And another genus, I think this is what I really like the most. This is Buxus. This is the genus Buxus. It was also a PhD project from Pedro Gonzalez from Origen. Buxus has some 40 species here in the Caribbean. There are two hotspots. One is Madagascar. The other is the Caribbean. Like in Europe, we have two. Here there are 40. And the question is also, looking at the Caribbean level of endemism, it's 70% endemism in the species. What was also interesting, because in the context of the Flora de Cuba program, there has been field work in Cuba for about 20 years. Many of the species actually have been described in the course of this project. So there was the taxonomic expertise, there was the material. Some of the species are extremely rare. And so we had to look at that. And I think what is also interesting, that's just some idea. We have been reconstructing the same way, and what is turned out that Cuba really was sort of the hub for species diversification, and apparently it went from Cuba to have colonized other islands. So the question is also what fuels speciation. This is what statistic analysis one can do, and one of the issues is really accumulation of nickel. So we have been looking at this. Most of the endemics are really confined to nickel substrate, and it could be really shown that there is an increased adaptation to nickel, not only tolerance, but hyperaccumulating nickel, and this is really fueled the speciation phase in this genus. Same patterns again, this is another genus, Cassiaria, where we again find multiple lineages of species. And this was the first study really to look into taxa that are more widespread, also in Colombia, in South America, in order to test species relationships, and species limits, which are sort of usually only have been looked at in islands or South America or Central America, and I think this is important to bridge. This is another tree. I'm not going into details. Rubiaceae. It's not. I would have thought orchids are the most species-rich group. It's not. It's Rubiaceae, the coffee family. And one surprise that came up, also looking at species level, there was the genus Taenus, rediscovered in the field in Haiti and the República Dominicana, and actually a new genus was found and described here. So this is a very brief summary of what we know. I think there is compelling evidence that Carlandia is not really an explanation for the majority of the flora. We also have an evidence that in the late Miocene, most of the radiation started. And we also have to look at, this is very clear, traits to understand really what the generation time effect is, or because the generation time effect, which says, okay, are there many is there a lot of time, and does that correlate with the number of species in an island or the Caribbean? That proves not to be very relevant. It's rather the traits, and well, adaptation to nickel is one, but there are many other traits, and I think that will be the work for the future, for the coming years to look at. And I think this is also, I'm just putting this as a last slide here on integrative taxonomy. So we need to think about the way how we're making taxonomy, how we're writing flora. And what was, yeah, I think one of the, Vanessa, you need to comment about this, essential things to make progress in the Flora project when you think about the young generation, PhD students, postdocs. They are not writing treatment because this is often like with a typewriter. It's rather creating from structured data that were also used in phylogenetic analysis, descriptions in an automated way. And this is also a tool that is used in the Flora project, the edit platform of taxonomy, which I think is something very important to really link academic research, taxonomic treatments, users' needs, and also to produce taxonomic treatment at a very good level of quality. And with this, I end, and thank you very much for this.
Muchas gracias, Thomas. Is there any questions?
Thank you, Thomas. It's a fascinating presentation. I have a question. Could you give us an idea of the range, possibly general range or extent of this type of work, either throughout the Caribbean or Central America in this region? This kind of analysis, is it happening across the region in other areas, or should there be? Maybe you could just comment.
Well, it's of course, as you have seen, there are model groups that we have selected. And looking, for example, at the genus Fuchs, it was continuously work 20-year project in order to get including treatment. Of course, we want to broaden. There have been contacts with a lot of institutions, but maybe we can bridge more. And as you see, there are ongoing projects. There are also funding. So it would be great to broaden that.
Another question? No questions. Well, a round of applause for Thomas. Thank you. Gracias.